Hi everyone, we're going to continue our discussion about genetic linkage, and we're going to talk about genetic mapping and how genetic maps can be built from linkage data. This material is found in chapter 14 in your text. The first question, the distance in map units between two genes on a chromosome is determined to be 18.6. This indicates what? Number two, when a red-flowered, oval-fruited plant was crossed with a yellow-flowered, long-fruited plant, the progeny were all red and oval. A cross of the red oval progeny with the yellow long plant produced the following. How far apart are the two genes? Number three, examination of test cross data in Drosophila showed that the two linked genes P and Q had a recombination rate of 0.05. And a third gene, D, had recombination rates of 0.5, 0.05, and 0.1 with P and Q respectively. What is the order and distances of these three genes on the chromosome? So Thomas Hunt Morgan was one of the pioneers of modern genetics. He identified X linkage, he identified that genes were found on chromosomes, and he introduced Drosophila as a model organism. What he noticed and what he proposed were that genes that were linked on the same chromosome but that were very close together would not be recombined very frequently. That's because recombination will occur at random spots along the length of a chromosome. And so two genes that are tightly linked will rarely have recombination. For example, this picture here shows a recombination event that does not mix up the big A and big B alleles. On the other hand, if genes are far apart on the same chromosome, here big A and big B, A and B are located at opposite ends of the chromosome, recombination is more likely to happen in between those two, and you will get more recombinant gametes. So the frequency of recombinant gametes is proportional sort of to the distance between the two genes. One of the students that was working with Morgan was Alfred Sturtevant, who became another leader in the field of genetics. He discovered while working with Morgan that recombination frequency could be used to map the genes along a chromosome. Here's a quote from Sturtevant's book. In a conversation with Morgan, I suddenly realized that the variations in strength of linkage, already attributed by Morgan to differences in the spatial separation of the genes, offered the possibility of determining sequences in the linear dimension of a chromosome. I went home and spent most of the night, to the neglect of my undergraduate homework, in producing the first chromosomal map. Amazing. Sturtevant showed that the frequency could be used to measure the distance between genes. Here's data from the cross that we looked at last time, where we crossed a wild-type fruit fly with one that had mutations for eye color and for wing shape. If you remember, it was a wild-type parent crossed with a double mutant. The F1 were then test-crossed. The F1 were wild-type, but were carriers of the two mutations. The test cross results showed that the original parents, the wild type and the double mutant, were overrepresented in the offspring. So instead of getting a 1-to-1 to 1-to-1 relationship, there is overrepresentation of the original parents and underrepresentation of the recombinants. What Sturtevant did was he counted up the number of recombinants. In this case, the number of recombinants is a 151 plus 154, or 305. He then counted up the total number of progeny, which in this case is 2,839. Then he simply divided the number of recombinants over the number of total progeny, which was 10.7%. 10.7% of the total flies were recombinants, were products of recombination. He defined this as in terms of a unit, or a map unit. And now we say that 10.7% recombination is the same as 10.7 map units separating the two genes. The more recombinants that are showing up in the test cross, the greater the distance between the two genes, because the more chance for recombination to happen. This is illustrated by this picture here. You can use this data to map genes against each other if you perform crosses. If you performed a cross between genes A and B, you would show that they're separated by a larger distance. If you did a cross between genes B and C, they were separated by a short distance. You would get few recombinants. And crosses between A and C would also give you a large number of recombinants. So that in that way you can map the distance between genes in map units. Data for different traits can be combined into a map. Genes X and Y, let's say, were 10 map units apart. Genes X and Z are 4 map units apart. But genes Z and Y are 6 map units apart. So by doing these 3 crosses, X with Y, X with Z, and Z with Y, you can build up a map. It would look something like this. We can show that X and Y are separated by 10 map units. X on one side and Y on the other. But X and Z are only 4 map units, and Z and Y are 6 map units, so the total distances are additive. 4 plus 6, of course, being 10. The first genetic maps of chromosomes were produced this way. Linkage groups can then be assigned. A linkage group is any group of genes that are linked together. A, B, and C in this case are all linked. Gene D and E are not linked to the first 3. However, D and E are linked to each other. So you would have 2 linkage groups in this scenario. Linkage group A, B, C, and linkage group D and E. Alright, that is all for today. I will see you guys in class.